Hello, my dear students, and welcome back. I know. Exams are just around the corner, but please don't you worry one bit. Your favorite tutor is here, and trust me, together, we are going to make this chapter super simple and honestly super fun. Are you ready? Let's go.
Okay, so let's imagine something. Think of this as your very own secret mission. You found a single, absolutely life-saving gene. Let's say it's the gene that makes insulin, to help people with diabetes. Now, one single PAPI is useless for making medicine for everyone. Your mission, should you choose to accept it, is to make lakhs and lakhs of copies of this one gene. So, how on earth would you do it?
Well, the answer, my brilliant little agents, is a kind of superpower. It's called Recombinant DNA Technology. Honestly, just think of it as the ultimate Control-C, Control-V for life itself. This is the amazing tech that's going to help us complete our mission and create all those medicines we need. Now, every superhero needs their special gadgets, right? Batman has his utility belt. We have ours. For our big mission, we need two very, very important tools. So let's get to know them. Pay close attention now because this is the most fundamental concept of the whole chapter. So what are our two tools? It's simple. We need a factory and a delivery vehicle. The factory will do all the copying for us, just churning out copies. And the delivery vehicle? Well, it's going to carry our precious gene and deliver it safely to the factory. In biotech language, our factory is the host cell, and the delivery vehicle is our vector. Easy, isn't it? Just remember, factory and delivery vehicle.
Now, what's our factory of choice? It's a tiny little bacterium you've definitely heard of, E. coli. You might be wondering, why E. coli? Well, because it's like a super fast printing press. Get this, it can double its population every 20 minutes. Just imagine, 1 becomes 2, 2 becomes 4, 4 becomes 8, and before you know it, you'll have millions of tiny factories all making copies of your gene. It's amazing.
Okay, so we have our super fast factory all ready to go. But wait, how do we get our gene inside it? We can't just push it in, right? For that, we need our special DNA delivery service, our vectors. So the most common delivery vehicle we use is something called a plasmid. The easiest way to think of it is like a tiny, super reliable delivery scooter. It's just a small, circular piece of DNA that's naturally found in bacteria, totally separate from their main chromosome. It is just perfect for carrying our gene right into that E. coli factory. And here we have it. This is one of the most famous delivery scooters in all of biotechnology. It's called PBR322. Everyone, this diagram is very, very important for your exam, so please look at it carefully. It's basically a map, a blueprint, showing all the important parts of the plasmid that we need for our work. So what makes a good delivery factor? Just 4 things. Think of the parts of a scooter. First, it needs an engine, which we call the origin of replication, or ORI, so it can start copying itself. Second, it needs a GPS, a selectable marker, which helps us find which bacteria actually took it in. Third, a special keyhole, a cloning site, which is the exact spot where we can insert our gene. And finally, it should be a small size so it's easy to handle in the lab. Engine, GPS, keyhole, and small size. Got it? Perfect. Now, our little scooter, the plasmid, is great for carrying small packages. But what happens if our gene is, really big and heavy? We can't put a huge Almira on a tiny little scooter, right? We're going to need bigger vehicles for that. And for those bigger genes, we bring in the heavy-duty vehicles, bacteriophages. Now, these are actually viruses that naturally infect bacteria. They are experts at injecting their own DNA inside a bacterium. So clever scientists thought, hey, why don't we use them as cargo trucks to deliver our large DNA packages right into the E. coli factory? Brilliant, right? And here are 2 very famous models of our cargo trucks. On your left, you have the lambda phage, and on the right, the M13 phage. They look a little bit like spaceships from a sci-fi movie, don't they? But they are expert DNA delivery specialists, and they're capable of carrying much, much larger gene fragments than our little plasmid scooters. Okay, my dear students, this table is super, super important. Please make a note. A plasmid scooter can carry up to 10 kilobases. A bacteriophage truck can carry up to 25 kb. For even bigger cargo, we have something called a cosmid, which is like a super truck, a hybrid of a plasmid and a phage. And for massive, massive projects, like mapping the whole human genome, we use jumbo jets like YACS, that stands for Yeast Artificial Chromosomes, which can carry up to 500 kb. Definitely remember these numbers for your MCQs.
So let's just step back and look at the entire mission blueprint. First, we use special molecular scissors to cut out our target gene. Then we paste it into our delivery vehicle, the vector. Next up, we send this vehicle into our factory, the host cell. And finally, we just sit back, relax, and let the factory run and make millions and billions of copies for us. It's that simple.
All right, is everyone feeling confident? I think so. Let's do a quick-fire revision round just to lock these concepts in for your exam. I'm going to ask some questions that are very commonly asked. Let's see if you remember. First question, nice and easy. What are the two most important things, the two key components we need for gene cloning? Think about our main story. What were the two main characters? That's right. It's the host, which is our factory, and the vector, our delivery vehicle. See, it's so simple when you remember the story. You'll never forget it now.
Okay, next question. We said we use E. coli as our factory, but why E. coli specifically? What's so special about this little guy? There are three main reasons. Can you remember them?
Yep. Number 1, it grows super fast, doubling every 20 minutes. Number 2, it's very easy to manage in the lab. No fuss. And number 3, scientists have been studying it for so long that we understand its genetics perfectly. It's just a very reliable and efficient worker.
And the final question for our little revision quiz. Think back to our delivery scooter analogy. What were the 4 essential parts that our vector, our scooter, must have to work properly? Exactly. You just have to remember the scooter. It needs an engine, the ORI, to get started, a GPS, the selectable marker so we can find it, a keyhole, the cloning site to put our gene in, and it needs to be a nice, small size so we can handle it easily. Perfect. You've got it.
And just like that, mission accomplished. Come on, give yourselves a big pat on the back. You have just mastered the core concepts of the host vector system in biotechnology. You guys are absolute superstars.
So if you're ever in the exam and you forget the process, just remember this simple mantra. Say it with me. Cut the gene, insert in vector, transfer to host, and let it copy. That's really all gene cloning is.
You now hold a truly amazing superpower. So I'll leave you with this thought. If you could copy any gene in the world to solve one big problem, maybe to create life-saving insulin, or produce a new vaccine, or even design a crop that can grow in the desert, what problem would you choose to solve first? Think about it. All the very best for your exams, my dear students. You are going to do wonderfully. I have full faith in you.